So this is just a review from yesterday before we finish our last half of notes. We do have a felony database of DNA fingerprints of any person who is convicted of a certain level of felony or higher. And we are now able to go back and look at maybe prior crimes that were never discovered who the perpetrator was. They can run any DNA from a crime scene in the felony database, and it will pick up a DNA. So this one, suspect one, matches somebody from the felony database. And this is just something that we're doing now. If you're convicted of a felony of a certain level or higher, you have no choice. Okay, your DNA becomes part of the DNA database. Another thing we're using DNA technology for is to find diseases or conditions. This is a whole bunch of people in a family, and this is a genetic screening for premature menopause. Premature menopause is usually when women go through the process where they stop having their monthly cycle and they can no longer get pregnant and bear children. And so early onset usually occurs in the 30s. So 3 members of this particular family would have this particular condition. So this would allow them to make arrangements if they wanted to have children. They could harvest eggs before the menopause happened so that they could still have children. These other members of the family do not have that gene because they don't have that stripe. This was the process they used to figure out how the cancer runs in my family, that it's a dominant cancer that is not found on the sex chromosomes, because 9 of my family members, including myself, we gave up DNA for Duke to be able to kind of figure this out in the study. So that's just some uses of DNA technology. Now, we're going to look at how transgenic organisms are made. This is the picture in your notes. And what I'm going to do is kind of explain how this process works. So the first thing we have to do, and this is genetic engineering or making transgenic organisms. Those phrases mean the same thing. First of all, we have to choose a gene we want to use. And the example I'm going to use is any one of us, if we do not have cystic fibrosis, you can take a Q-tip swab the inside of your cheek, and then the scientists can remove the correct cystic fibrosis gene from you. And they can use restriction enzymes, which are like those scissors, except they're specific. They will only cut DNA at a specific point. And they cut our correct cystic fibrosis gene out of our chromosome. Then they use a restriction enzyme to cut that gene out. Sorry, got a little bit ahead of myself. So here's the gene that's been cut out of a healthy person's DNA from a cheek swab. Then they take a plasmid and they use the same restriction enzyme on that plasmid, and they make what's called recombinant DNA. And remember, the root word of this is recombine. Combine means you're putting stuff back together again. What does recombine mean? Okay, in recombining, you're taking something and putting it back together, but differently than you had it done before. So this is a bacterial plasmid, which only bacterial prokaryotic cells have, and it now has a human eukaryotic gene in it, which is the cystic fibrosis gene. Okay, so this is the recombinant DNA. It's been recombined. Then we insert this recombinant DNA into bacteria, and then we let this bacteria make a lot of babies asexually. So are the DNA of asexually reproducing cells, is the DNA similar or is it identical? Okay, it's identical. So these 2 cells are identical to the parent cell that made it, and bacteria will reproduce themselves every 20 minutes. And then we have these cells, and in this particular case, these cells will make a protein, whatever this gene of interest is that we inserted in there. So the example that I used here is cystic fibrosis. So here is a child with CF, And this person is actually putting through this tube genetically engineered viruses. So the picture that I just talked about, this is genetically engineering a bacteria. But we can use the same process, and instead of putting it in a bacteria, we can put it in a virus. So how does that work? Well, this girl, she has a clothespin on her nose because you don't want those viruses getting out of her mouth. You want it to go down to her lungs, so she's inhaling these viruses. Well, what happens when this transgenic virus gets to her cells? Well, here's the virus, and here's the viral DNA with the human correct cystic fibrosis gene inserted into it. These little red knobby things give it permission to enter our cells. So here it is, entering the cell. It floats down, and it injects the DNA into the nucleus. Here's the nucleus, and it uses one of these pores, which is what mRNA uses to get out of the nucleus. It uses the pore, and it inserts the correct gene. And then at that point, this particular cell is cured. And the reason why this girl is going to go in for treatments regularly her entire life is because every time the cells in her lungs reproduce themselves, they do reproduce that correct cystic fibrosis gene, but some of the cells don't get genetically engineered. Some of the cells, by pot luck, the virus will not get all of the cells. So some of these cells, this cell may get genetically engineered 100 times, whereas the cell sitting right next to it never gets engineered at all. But by using this method, we have increased the lifespan of people with CF from 18 years old, and think about it, how close are you to 18? To 34, roughly 34 years old. So instead of dying in a year to a couple of years, you would be able to live until you were 34. So it doesn't cure everything. This process only cures the lungs. It does not include all the cells on the digestive tract. This virus is actually killed off by the body's own immune system before it can get into the intestinal cells. So we still have some time to go yet on this, but it is a process. We are working on it. Now, so how do we know where the correct cystic fibrosis gene is? How do we know how to make all these transgenic viruses? How can a transgenic virus make human insulin? Well, it's something called the Human Genome Project. Okay, so we're going to actually look at a video. Whoops, here it is. Let's look at this video which explains it.
Thanks for stopping by. Today we are talking about the Human Genome Project. Before we discuss the actual project, I want to define what a genome is. So a genome is the complete set of genes in an organism. In the Human Genome Project, that organism is, of course, the human. There are several scientific techniques that made it possible to start and complete the Human Genome Project. These include DNA sequencing, polymerase chain reaction, and electrophoresis. The Human Genome Project was essentially an international research project to map the entire human genome, every section of DNA in a human cell, which is a lot, 3 billion base pairs, in fact, and they've mapped all of them, which took 13 years to complete. The project determined that there are about 20,500 genes in the human genome, and it also discovered the order and location of these genes. It didn't, however, identify what each gene does, and that research is ongoing, and there's still a lot to learn.
Okay, so how do we know there's 20,500 genes in the human genome? Well, because we did this project where we wanted to identify where all the genes are in all of our 46 chromosomes. This is where the Human Genome Project is stored. All the information, all the genes, literally the base sequences for all the genes in a human are stored at a computer in the Northeast United States. So why did we do this? It took 13 years, but why did we do it? Well, for gene therapy. Okay, we wanted gene therapy, which means like what we're doing with CF. You have the people with CF, they inhale transgenic viruses. So those are viruses where we've taken out the DNA that causes us to get sick and we put in a human gene, and then the viruses go and infect our cells with the correct gene. That is gene therapy. And we are trying to do this. Okay, so let's finish this.
It continues to be a benefit to society as a massive information source for researchers to draw on and to continue their advances in both science and medicine. I hope this brief explanation has piqued your interest, and if
Okay, so for science and medicine, to help cure genetic diseases for gene therapies. Okay, so that's why we are mapping the human genome. Okay, so this is why these are the notes for the honors. You have this picture, your steps are outlined, and then it's talking about the Human Genome Project. Okay, standard, y'all have the same diagram. Okay, you are not required to know the actual order here, but it's got some information for you as well, and then what you do need to know. Okay, also you have your Human Genome Project as well. So we are doing this to try to cure people of genetic diseases. What we need to look at next is clones. So that's the last bit of the notes, cloning. So let's look at clones. All right, clone is an identical genetic copy of an organism. Genetic identical copy of an organism. Now, here's your words, and you have that right here, and I'm going to explain it using this picture. Okay, so you've got words and picture. This was the first mammal ever cloned. We called her Dolly, and she was a white sheep. So we took a black sheep, took out the egg, an egg from one of her ovaries, sucked out this haploid nucleus, because remember, gametes are made through meiosis, and they have a haploid nucleus. We sucked out the nucleus, and we took a diploid skin cell nucleus from our Dolly, our white sheep. And we inserted the nucleus into that egg cell. We processed it. We then put it back into this black sheep and let her carry the baby to term. When she gave birth to Dolly, an identical genetic clone. Now, why do we use a black sheep to give birth to a white sheep? Well, this black sheep was homozygous dominant. Does homo mean same or different? Okay, so this sheep had two capital black alleles, big B, big B. White is recessive. It always is in all animals. And so it's not genetically possible for a homozygous dominant black sheep to give birth to a white sheep. Why? Because white is recessive and this sheep has two capital Bs. So they wanted to be sure that the sheep that was born was not from this black sheep. Okay, now, so that's what we did. We cloned Dolly, and here it made Time magazine. So there's Dolly and her clone. Now, a clone is born as an infant. Okay, so I have this. A clone is an animal born as an infant, a baby. This sheep did not give birth to a full-grown sheep. If it did, it would have killed her. So the clones are always born as babies, as infants, and then they have to grow up. But there was a problem. When Dolly's clone was 2 years old, she went blind. She had arthritis so bad that she couldn't walk, and Dolly's clone died a little over 2 years later, at 2 years old. So what happened? Well, it turns out that DNA has an age stamp. All of our DNA has an age stamp on it. Well, what does that look like? This one, nope, this right here. Okay, so this is a bunch of chromosomes. You see the funky looking X's, and at the end of each chromosome is something called a telomere. And what happens is it's kind of like a slice of bread. If you get a brand new loaf of bread, you open it up and you take 2 pieces out, you still got a pretty big loaf of bread. But by the end of the week, You keep taking 2 pieces out, 2 pieces out, 2 pieces out. By the end of the week, you're out of bread again. That's exactly what happens when animal chromosomes replicate through mitosis. Every time they replicate, this is a beginning in a baby. Look at all the telomeres. Each little like tick mark here is a telomere. As the cell undergoes mitosis, all cells but brain and muscle cells in the human body undergo mitosis to make identical clones. A piece of one of these telomeres is taken off. And then as you age, notice what happens to the amount of telomeres at the end of the gene, at the end of the chromosome. They get down to where there's no more left. That is when enough of the cells in a particular organ reach this state, that's when that organ fails. So some people, as they get older, they have kidney failure or liver failure because they have used up all of their telomere, which is a protein. So we found out that poor Dolly, when Dolly was cloned, her DNA was 2 years old. So the day that Dolly's clone was born, even though it was born a baby, Dolly's clone's DNA was 2 years old. So that when Dolly's clone reached physical age of 2 years old, her DNA was the physical age of a 4-year-old, and 4 years is when she'd die. So her clone died early. This is why it is illegal to clone humans. It's one of the reasons why it is illegal to clone humans. To clone a human, we would have to take that human's DNA the day of his or her birth to clone. Because if you were to take your DNA, and let's say you're 15 years old, then the day your clone is born, your clone's DNA is already 15 years old. So that clone's life will be shortened. So that's part of the reason why it is illegal to clone a human. And there's religious issues as well, dealing with a soul, and does an organism have a soul? So there's other religious reasons as well, but it is illegal to clone a human. Well, once scientists realized this, they created these 2 mice right here, and they're called Adam and Eve, and they're rats. What they did an experiment where they injected their cells. They actually gave them a diet where they had telomeres. They were injected with telomere, and they were able to, their body was able to replicate. So even though their cells were undergoing mitosis and the telomeres at the end of their chromosomes were deleting, because they had the ability to make new telomeres, they were able to maintain kind of like that full loaf of bread, which is, that picture right here. Adam and Eve are more than 15 years old now, and they're still alive. They're still in their prime. They're still having babies. In other words, they have not aged. By giving them this protein, they have stopped the aging process. So yes, you could call this, quote, the fountain of youth gene. Cells lose the ability to make telomeres once we're born. Can't make it anymore. The normal lifespan for mice is 2 years, and rats. These rats at 1 year old, the prime of their life, they were given and are still being given these telomeres so that they are not aging. It is illegal to make this for humans, and it is illegal for humans to make for a lot of ethical reasons. Who gets it? Who gets to live forever and who doesn't? Ethical issues as well, religious issues. Okay, so let's look at, this is cloning the woolly mammoth, because making clones is expensive.
The woolly mammoth hasn't been around since the last ice age, 10,000 years ago. Well, it won't be roaming modern-day Manhattan tomorrow. Scientists say they just got a lot closer to the day it could.
For the first time, it is not entirely impossible anymore.
The discovery by a team at Penn State University was made possible because of ancient hairballs left over from woolly mammoths that might allow scientists to reconstruct significant portions of the mammoth's DNA and could conceivably, for the first time ever, resurrect an extinct species. Scientists would take DNA from the mammoth's hair and use that to alter DNA from a modern-day elephant egg. The new eggs would eventually be implanted in a living elephant, carried to term, and delivered as a living, breathing mammoth.
There's probably no limit other than our own imagination as to what we can do with these data.
Ross McPhee is a curator at the Museum of Natural History in New York. And while Jurassic Park coming to life may sound like a great idea, McPhee says, just like in the movie, just because we can doesn't mean we should.
Are we going to bring them back just for laughs, to show them off in carnival kinds of contexts? Are we going to take care of them? Where are we going to keep them?
Researchers say the practical applications don't end with the woolly mammoth.
We immediately want to apply this knowledge towards saving endangered species that live at the brink of extinction today.
Actually, bringing a mammoth back will take at least 10 years and at least $10 million. But for now, what a wonder to even think about. Jeff Glor, CBS News, New York.
Okay. So, again, just because we can do a thing, this is the ethical issue of DNA technology. Just because we can do these technologies doesn't mean we should. Now, the last video I have for you is a real shocker. There is a man who is trying to clone humans. It is illegal in all but 3 countries for religious reasons, for population issues with, humans running out of space, and some other issues. So let's take a look at this video.
Eerie blue light of his secret lab, one of the world's most controversial scientists shows off his creation, cloned human embryos that are genetic carbon copies of their father.
Oh, my babies are doing well. They look beautiful.
Filmed for a TV documentary, Dr. Panagiotis Zavos implants 11 of the embryos in 4 women, one of them British, in the hope of producing the first cloned human being. The procedure is a criminal offence in the UK, but not in this undisclosed Middle Eastern country.
I think we have 3 very good embryos that could be in utero today, and that if implantation is successful as well, and the pregnancy is maintained, as we say in the US, we have a home run.
Dr. Zavos also reveals he's produced cloned embryos from 3 dead people, including Katie, who died in a car crash. He injected her DNA into a cow egg, though he hasn't implanted the animal-human hybrid. British scientists have condemned his work as reckless. Cloning of animals, such as Dolly, has shown the technique carries risks of severe congenital abnormalities.
I find it A, incredible, and B, disgusting, because he's sowing false hope to begin with. So the individuals who are involved in this experiment, and that's what it is, an experiment, may well not realise that the outcome is almost certainly going to be negative. And to put couples through that, in my opinion, is unethical.
Dr. Zavos adapted a standard cloning technique for his human patients. He first took an egg from the mother and stripped out the nucleus containing 99.9% of her DNA. He then took a skin cell from the father and extracted the genetic material. That was injected into the empty egg and given a jolt of electricity to form a cloned embryo. Within a matter of days, the embryo would have grown into a ball of 32 cells, which Dr. Zavos implanted into the mother's womb. So far, none of the attempts has led to a viable pregnancy, but this infertile Canadian doctor, who wants to remain anonymous, says the technique is his only hope of having a child.
Pretty much decided that probably I would be the first one to be cloned, but then, if it worked, then we might go on to have another child.
Despite almost universal condemnation, Dr. Zavos has vowed to continue his work. He says more than 100 patients have contacted him in the hope of having a cloned baby. Thomas Moore, Sky News.
Okay, now for the what Zavos was talking about was he took an egg that was before meiosis occurred. So when he talks about the egg that contains 99% of the female's body, right here, he took the egg before meiosis occurred. So when he took that nucleus out and put the male's diploid nucleus in, this was still an undeveloped egg, which when he jolted it with electricity, which is what they actually do in IVF for people who are trying to have pregnancy, you do have to kind of start that process. Then the egg was like, oh, okay, I need to go and I'm going to start making a baby. So he used an unmature egg, which is why he said it contained 99.9% of the mama's DNA. But that's just for him, his process. But there are a lot of ethical issues. So if this particular man who is here has a clone, well, in religious terms, does his clone have a soul or not? If they're a genetic identical clone. What if the clone has abnormalities, or the DNA is older and the clone's going to have a shortened life? Will the clone have the same rights as this man here? He technically would have the same social security number, the same, like, how does this person interact in our world? And why would we do this? Would people make clones just so in case they had heart disease, they would take the heart out of their clone and kill that person and then put the heart in themselves? I mean, what are we going to do here? There's a lot of ethical issues that go with this unit that we really can't. We in our country, it's democratic, so there are laws, like making a human clone is illegal in all countries but 3. But there comes, how are we going to behave with all this? And that's part of this issue. And you will have an assignment at the end of this unit, which you'll give you some chances to do some writing on it.